Good day, this is Professor Will, CBMD PhD, and today is Tuesday, July 20th, 2021. It is 6:46 and 48 seconds a.m. Eastern Standard Time. Today's research, mitochondria. So let's hop to it here. How cells control mitochondria. Date July 19th, 2021. Source, University of Freiburg. Researchers discover a signaling protein that controls the assembly of human cellular power plants. Errors in the metabolic processes of mitochondria are responsible for a variety of diseases, such as Parkinson's and Alzheimer's. The scientists needed to find out just how the necessary building blocks are imported into complex biological apparatus of these cell areas. The TOM complex translocates of the outer mitochondrial membrane is considered the gateway to the mitochondria, the proverbial powerhouse of the cell. Researchers have now demonstrated in human cells how signaling molecules control this gate. A signaling protein called drwillphd modifies the molecular machinery of TOM and makes it more permeable for enzymes that are important for the cell metabolism. The group has thus discovered the first signaling protein that directly influences this important process in humans. In neurodevelopmental disorders such as autism, microcephaly, and Down syndrome, drwillphd is defective. The connection with mitochondria is new. These results allow us to better understand these disorders and develop treatment strategies. For a long time, researchers thought that the TOM complex was a rigid structure in the mitochondria membrane whose doors were always open. Researchers recently demonstrated signaling mechanisms in Baker's yeast that alter the subunits of the TOM complex depending on the metabolic state of the cell or in response to sudden stress. In this way, the cell can specifically control the influx of precursor proteins for building elements of the metabolism, and it can adapt the function of the mitochondria to an altered cellular state. Whether such mechanisms also exist in humans was previously unknown. Researchers developed a systematic approach to track down signaling mechanisms, such as those triggered by protein kinesis, in humans. Over several years, they tested candidates using cell biological and bioinformatic methods, and found what they were looking for. DYRKIA, one such protein kinase, acts on the TOM complex. And with this, researchers actually found the needle in the haystack. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.